Hello, good afternoon. Let's get started. We are going to finish lecture 7 today. And I'll tell you about next week, the schedule and the lab and everything. So stay tuned for that. Now, last class, we learned how to do transcription and translation. And I think this is the slide that we left off at, right? This one, example 3. I told you to try example 3 on your own at home. And hopefully you had a chance to do that. If not, that's okay. We're going to take it up right now. So we have the coding strand here. Coding strand. Oops, something's not working. What's going on here? Let's try that again. We have the coding strand, which is the strand that is containing the instruction to make the protein. And so if I want to do transcription, I can just go ahead and do it. What is this thing doing here? That is so weird. Let me see if I can get that out of the way. So we just copy it. AUG, of course you have to change the T to U because there's no thymine on the RNA. And then you can put a dash here, ACA, UUA, CUA, GCA, GGC, and then, sorry, running out of space here, but ACU, right? Then we can go ahead and find out the protein sequence. AUG is the start codon, which is MET. Now, someone asked me, like, is it possible to have an MET somewhere within the protein? And that's the answer is yes. Okay? Even though the start codon is MET, but it doesn't mean you cannot have another one later on in the middle of the protein, right? You can have another AUG code. Just like when you write in English, the first letter has to be capitalized, right? First letter of the sentence has to be capitalized, but it doesn't mean you cannot have a capital again later. Like if you have a name, if you have a place, right? Then the capital is going to happen again, and that's kind of like that. ACA is going to be threonine. UUA is going to be LEU. Right? CUA is another LEU. GCA is ALA. And then GGC is going to be glycine. And ACU is going to be THR. Now this one doesn't stop because maybe this is not the end yet. Maybe it keeps on going, right? Like just because the last one is not a stop doesn't mean you're doing it wrong. It's just that it means the sequence is not finished yet. Okay, so that's what we did last time. Let's quickly do a poll question here. If a codon contains 4 nucleotides instead of 3, what is the maximum number of amino acid it can code for? What do you guys think? You might need to pull your calculator out for this one. So 4 nucleotides. We have 4 choices in the first blank, A, C, U, G. Another 4 choices, another 4 choices, another 4 choices, right? So if you multiply all these together, you will get 256. So this sort of explains why the codon, it's 3 nucleotides. If it was just 1 nucleotide, then we only have 4 choices. That's not enough to code for 20 amino acids. What if we have 2 nucleotides to make up the code? That's 4 times 4, which is 16. 16 codons is also not enough to code for 20 amino acids, right? So that's why it is a triplet. That gives us 64 codons in total, encoding 20 amino acids. And there are some redundancy, right? As you will see later on today, that redundancy is very useful as a protective mechanism against mutations. All right. So now we're going to move on to translation. We're going to talk about how translation takes place. And the whole process has 3 kinds of RNA that are involved, some of which you have already seen before, and there are some new ones as well. So messenger RNA, by now, you know that is a copy of the DNA, a copy of the gene that we're interested in changing into the protein, right? And basically, it gives you the instruction to make the proteins. So this is, you can think of it as a recipe. Okay, the recipe. And then you have the rRNA. The rRNA is the one that is actually going to be it's the component of the ribosome, and it is the part that is going to basically make up the protein. It's going to connect the amino acids together to form the protein. And so in our analogy, it's going to be the chef. Oops, going to be the chef, the one that's like cooking the recipe. But, you know, it has to follow the instruction in the recipe, and it's gonna make the protein. It's gonna make the dish. And then we have this last one here. That's the tRNA, which you have seen in lecture 2, but then I haven't really give you too much information about it. The T stands for transfer RNA, and the transfer RNA helps bring the correct amino acid to the ribosome, right? So you can connect them together. So you can think of this as the helper, okay, the assistant, to bring the ingredient to the chef so that the chef can cook the recipe. Understand? Messenger RNA has the instruction to make the protein. tRNA brings the correct amino acid, and the ribosome is the rRNA, which makes up the ribosome. That's the one that is responsible for connecting the amino acids together. Okay? Any question? All right, I'm gonna give you a note. I will be like erasing things and moving things around quite a bit. I don't know. I mean, you can copy along the way, but just know that I'll be moving things around, and you might have to reorganize the note later on. It's also okay to not copy if you just want to like sit back and listen, and then once you listen to it once and you understand where the note is going, you can watch the recordings later and copy it. Okay, so let's take a look at tRNA. tRNA is, again, stands for transfer RNA, right? This is the RNA that will be bringing the correct amino acid to create the protein. So give me a second here. The tRNA is RNA, so it's single-stranded. But what it can do is it actually can fold back on itself, right? It can fold up and form a shape that looks like that looks like a clover leaf, kind of like a star, if you would. Okay, and the reason why it's able to fold back on itself like this rather than just, hanging loose as one strand, is because part of the strand is able to form complementary base pairing with itself. Okay, so there are some bonds here. There's some bonds here. It helps to stabilize the structure. Okay, so I will give you an example. This is again RNA, right? So it has A, C, U, and G. So let's say this over here is A, C, C, U, or something. Then at another part of the strand, it just happens to be U, G, G, A. So when it loop backs on itself, is able to form these complementary sections. Okay, complementary pairs at different parts of the tRNA help stabilize the structure. This part you can copy. Okay, this was no problem. I'm not going to erase this. Okay. And then same thing here, right? You will have some like complementary base pairing going on, and it creates this like clover leaf looking club shape structure. Let me just give myself more space. Now at one end over here, we have a site. This is called the amino acid attachment site. Right, this is where we're gonna put an amino acid, and I can put any amino acid on it. Let me just take out my codon chart so I know I'm doing this right. Let's say this one is like this one is like SER. Okay, SER. A different transfer RNA is going to be carrying a different amino acid. Okay. So how does the tRNA knows which amino acid to carry? Well, at the bottom of our tRNA, at the bottom right here, there is a triplet of special code. Okay, and this special code is going to be called the anticodon. Let me just get this right here. Give me a second. That would be like AGG. So this right here is anticodon. Okay, anticodon. The whole thing, of course, the entire tRNA is made up of just A, C, G, and U, but specifically right at the bottom here, there are 3 codes that are important. That is what we call the anticodon. So what does the anticodon do? Okay. I'll write down here. Anticodon is going to be complementary to codon on messenger RNA. And the anticodon will also determines which amino acid the tRNA carries. Just copy it down first, I'll explain. All right. Now this next part, I'm gonna be moving things around and erasing things. So maybe you just want to sit back and watch. All right, so this here is gonna be the messenger RNA. Okay, 5' to 3', messenger RNA. And I'm just gonna make some code here. Okay. AUG, UUU, you know, UCC, ACG, and then UAA, something like that. All right. Let me just make this smaller, give myself more room. I always run out of room when I make a note. Okay, so there it is. Okay. Now we're gonna have our ribosome. Let me choose the ribosome here. Okay, so the ribosome is gonna basically clamp on here with the large and the subunit. Okay, remember, ribosome is made up of rRNA as well as proteins. All right. So can you look it up? Can you look up AUG? What does AUG code for? You should know this by now. We've done this many times, but humor me. Tell me what AUG codes for. Anybody? Yeah, thank you, Sarah. It goes for MET, right? It goes for MET. So in the cytoplasm, we're gonna have a bunch of tRNA, and I'm just gonna draw it like this. And so there's a tRNA here, there's a tRNA here, there's a tRNA, and I'm just gonna draw a couple more. Okay. So this one, let's say this one carries the MET. And then this one carries, totally making this up, LYS. This one carries PHE. This one carries, hold on, I just gotta make sure that this is correct. SER. All right. So the question I'm trying to give you the answer to is, like, how does your body know which of these is suitable to put for the first codon? Well, it turns out each one of these tRNA I told you has an anticodon at the bottom, right? So this one is gonna be UAC. This one is going to be, sorry, I'm totally making this up on the spot, so I have to check the codon. I have the codon chart on the other side. I just need to make sure that I am doing this correctly. So this one is like UUU. This one is like AAA. And then this one, I think I did it already, is a AGG. Okay. So try to follow me here. Again, you don't have to really draw it at this point. Just watch. So let's say I take this guy, right, like in the body. This is just floating around in the cytoplasm, and then it try to goes here. Right? I try to come here. Is that gonna fit? Well, it's not, because if you look at the codon, which is the black, and the anticodon, which is the green, they are not pairing up properly, right? They are not complementary. And for that reason, if it's not complementary, it won't fit. So this one will just bounce off. Okay. What about this one? What about like this PHE? Maybe it's this tRNA that's carrying PHE is going to try to come here. Is that going to fit? Well, again, you have to look at the anticodon and the codon. Are they complementary? Are they pairing up? Obviously, is not, right? So that's going to go away. And then what about the next one here? The next one, the one carrying the MET, right? Is that complementary? Right. Well, if you pay attention, A and U, they pair with each other. U and A pair with each other. G and C pair with each other, right? So if they are complementary, it fits, it clicks, it's like puzzle. It fits together, and that's how your body knows this is the right amino acid. Okay? So this guy is basically gonna come and drop off the MET. So I can't really show you this part. Just imagine in your head, okay? Brings it over, puts the MET there, and then it's gonna go away. So after it puts the MET, oh my goodness, after it puts down the MET stays. Okay, and now our transfer RNA is empty. It needs to reload itself. Right? So there are a bunch of amino acids that's just floating around in the cytoplasm. It's gonna have to find another MET, and this one can only be carrying an MET, right? It's not gonna be carrying anything else because the anticodon, as I told you, determines which amino acid it's gonna carry. Okay? So pause for a second and see if you have anything you want to ask me. Okay, so next we have UUU. Okay. Can you guys, without looking at a codon chart, how does the amino acid stick to it? That's a good question. The ribosome actually has 3 slots for it. It's like almost, you know, those wooden toys that kids have, like have a cow or something, they try to fit into like whichever slot. It's kind of like that. Okay, there are 3 slots in the ribosome, and then you just put the MET in there. That's the short version. Okay, the actual detail we don't have to concern ourselves with. Now, without looking at our codon table, do you know which one of these is going to come? Right? Is this the right one? Is this the right one? Is this the right one? Like, which one is the right one? Let me make this full screen for you. Which one is the right one, huh? Which one's going to come? 15 said PXG, that's right. Right? Look, this is going to come, and AAA is going to match with UUU, right? So it's going to come here, and then it's going to basically deposit the PHE, and then the ribosome will form a link, a peptide bond. And then the other one is just going to flow off, right? It was AAA before. And now this one's going to have to find a PHE. Okay? And so on and so forth, right? Then the ribosome is going to basically move forward, shift over, and then the correct tRNA is just going to keep on coming and coming until the end, until you have the stop codon. Okay? There are no corresponding tRNA for stop codon. Okay, so UAA, UGA, UAG, no tRNA for that. Once you get to the stop codon, everything just pause because you're waiting for that tRNA to come, but it's never going to come. And that's a signal that tells the ribosome, that's it. You finished copying. You finished making the protein. You can stop now. And then the ribosome detach, the protein detach, and then we're done. All right. So let's go back to the PowerPoint slides and see if the pictures in the slide will give you a better understanding. Okay, so like I said, this is the tRNA. They carry the amino acid. And how do you know what amino acid it carries? Well, you have to look at the anticodon right here. Okay. But the chart, the table that we have, right? This table is for the codon, is not for the anticodon, right? So that's why when you when you're given a tRNA, you have to convert the anticodon into codon first, and then you look it up. If at any point on a test or on a quiz, you find yourself looking at the looking up an anticodon, you're doing it wrong. You should never be looking up anticodon on the codon chart, the table. Table is only for codon. Right? So let's take a look here. Number one, step one. Amino acids and tRNAs flow freely in the cytoplasm. tRNA are just always there. The amino acids, they come from the food that you eat, right? You eat proteins, you break down the proteins. Now you have amino acids available to build whatever you need to build. There is going to be an enzyme that helps connect the amino acid to the correct tRNA. So help me out here. This tRNA is empty. Maybe it just delivered its amino acid to a ribosome and now it needs to reload. And the anticodon here, the anticodon is GAG, GAG, okay, GAG. What would the correct amino acid be? Can you find out for me? Like if I gotta load this guy up with an amino acid, what should it be? How do you find that? Yeah, and how do you know that, Sarah, if you don't mind sharing? How do you know it's LEU?
I'll look on the chart.
And what did you look
of
up
the
on the chart?
codons.
What did you look up on?
And in the second row.
No, yeah, I understand that. But what code did you look up? What's the letter that you look up?
Okay, I look CUC.
That's right. Right. You're supposed to look for CUC. Thank you. This is the anticodon. And then the codon that's going to be on the mRNA, right? That matches up is CUC. And this is the codon. So you're going to look up CUC on the table. What is CUC? CUC. That's LEU. So this one is going to be carrying an LEU. Is that okay? Justine, you okay now? Do you understand why it is not?
Yes, sir. Like I
Okay.
just look at the wrong spot.
Yeah,
in
that's
the
okay.
column.
Yeah. Everyone else okay? You're gonna see this on the quiz. Okay. So then an enzyme will come and help load up the amino acid onto the tRNA. And then the tRNA will dock as in like will park into the ribosome if complementary RNA codon is present on the ribosome. Okay, so complementary. The anticodon on the tRNA must be complementary to the codon on mRNA. Okay, it must be, nice fit. If it doesn't fit, if it's the wrong pair, it's just gonna bump in and then it's gonna go away. Exactly like, doing a jigsaw puzzle. Sometimes you try the piece and it doesn't really quite fit and you just throw it away, get another piece and try again. Okay, so this keeps on going until you get the right piece, except it happens really fast in the cell, right? So if you get the right piece, then the amino acids join together to form a polypeptide. Right? So this thing has probably been building for some time now. I mean, like I can only imagine, like, there are a few other amino acids here, and at the very beginning is the MET, and then there is a bunch that you keep on adding onto it, right? And then the MT tRNA will go back, and let me just write this down for you. This is gonna go reload. Okay? Reload the amino acid. They're gonna get another one. And then they're gonna, if you need that amino acid again, they're gonna come around. They don't have to come back to the same ribosome. I mean, there are hundreds of ribosomes making proteins at any given time, right? So it might deliver it to another place. And then this will continue as the ribosome move forward until you hit the stop codon. I'll read this to you. When the ribosome reaches the stop codon, no tRNA can base pair with the codon on the messenger RNA. It doesn't exist. There's no such tRNA. And then the RNA and the newly synthesized proteins are released. Okay, so the protein fall off, and then we're done. So remember, the ribosome has large and small subunit. It will separate, and now it will find another messenger RNA to clamp onto. And then the process repeats itself. Okay. Please try this question. I want as many of you to participate in the poll as possible. So I know you know how to do it. Like I said, it's going to be on the quiz. Okay, I got 5 answers. Anyone else? Okay. Well, this is not looking good, to be honest. Only 2 people got it right. All right, let me try to explain this again. All right, here we have our tRNA, the one that looks like a clover leaf. They actually do look like this. And then it has an anticodon of AAA at the bottom. This is anticodon. And then the question is asking, what kind of amino acid is it carrying, right? So what should you be looking up on the table? Should we be looking up AAA? Absolutely not. I told you already. AAA is anticodon. If at any given point you are looking up anticodon on the codon chart, this is called the codon chart, you are doing it wrong. Okay? Maybe you should write with Sharpie, don't look up anticodon and then stick it in front of you when you do the test. All right, so I shouldn't be looking up anticodon. What should I be looking up instead? Well, AppNet said UUU, that is absolutely correct. You're looking for the codon. Okay, you're looking for UUU. And what does UUU code for? If you look it up on the codon chart, UUU codes for PHE. Are we together? Let's try another one, okay? Let's try another one. Let's say I have another guy here. Okay, let's say this is CUC. All right. Tell me what's the amino acid. Please do it. Okay. Again, I want to see your answer. Type it in the chat box. I did this with my in-person class yesterday. The students were not allowed to go on break until they show me the right answer. What are we looking for on the table? What are we looking for?
GAG.
We are looking for GAG, that's right. And if you look up GAG, the answer is GLY. Okay? If you don't understand this, please ask question now, okay? Don't try to don't think like, oh, I'll just look at it later and I'll figure it out. You got to figure it out now. Otherwise, you're going to get confused for the rest of the lecture. Any questions? Please re-explain. Which part? The whole thing? Okay. Do you know what a tRNA is, Tahib? Can you explain to me what a tRNA is? Based on what your understanding. What is a tRNA? What is this? What does it do?
Said it transfers RNA.
Yes, but what does it do?
Mm,
It is transfer RNA, but what does it do? Anyone else want to help out here? What does the transfer RNA do? Patrice, can you help out? Maybe. What does the tRNA do? I'm just randomly picking people on the list. Justine say building block for amino acid. Not quite it. To connect them, also not quite it. I'm sorry. Okay, maybe I'm not doing a good job explaining. Okay, write this down if you don't know the answer. Transfer RNA brings the correct amino acid to the ribosome. It doesn't do the connecting. It doesn't do the building. The rRNA, the ribosome, does the connecting. Okay, the tRNA is like the helper. It brings the ingredient. And then the ribosome, like I said, is the chef, right? The one that's going to be like making the thing. Okay? tRNA brings the amino acid to the ribosome. Now, at the bottom of the tRNA, we have 3 letters. The 3 letter is called an anticodon. Anticodon. How does it know whether it's bringing the correct amino acid or not? Well, the anticodon has to be complementary to the codon. I feel like I'm just writing the same thing again. But the codon is on the messenger RNA, right? It's on the instruction. So if I want to figure out what amino acid it is carrying, I need to look up the codon on the table. Look this up on the table.
Sir, is it not GLU?
What's that again?
Like GAG. When you like look it
GAG?
up on the codon table, is it not gonna be like
If I look up GAG, it's GLU. Yeah, you're right. GLU. Thank you.
Okay, thank you.
Okay. So if I give you an anticodon, you have to change it to codon first before you look it up. Questions?
How do we change to codon?
Complementary, base pairing rule. C goes with G, A goes with U, right? Okay. Here, I'll give you another one. CAU. If this is the anticodon, what's the codon? Give me the codon, Tahib. or anyone else?
GUA.
GUA, because C pairs with G, A pairs with U, and U pairs with A, right? And again, that's the one that you're going to be looking at. All right. So there are 4 levels of control of gene expression. I told you already, every single cell in the body contains the same genetic information. They contain the same set of ACTGs, okay? But you don't turn on everything. Depending on the function of the cell, you're going to switch on a set of genes that will be related to the function of the cell, okay? So how do you control what genes to turn on, and you know, when you do make the protein, how do you control how much of it to make, right? So it turns out the type of control can happen at 4 different levels. It can happen at the transcriptional level, which literally means you either transcribe the gene or you don't. If you don't want to make the protein at all, then you don't even do the transcription. If you don't do the transcription, then there is no protein, right? So your body can choose which one to transcribe and which one to not transcribe. Once you do the transcript, you can do post-transcriptional modification, right? You can change how long the tail is, the poly-A tail that I taught you last class, and that will help determine how long the mRNA is going to remain stable for. Okay, so you can regulate the length of time that the mRNA is going to be available. You can remove the introns and join the exons together. You can, in some cases, you can actually choose which exon to join together. Let's say you have exon 1, 2, 3, 4. You can connect 1, 2, 3, 4 together. You can choose to just have exon 1, 3, and 4. Or you can just have 2 and 4. So that gives you additional combination to change how the protein is built. Even after you build a protein, even at the level of producing the protein, when you're doing translation, you can control how fast and how much of the protein you're making. Do you make a lot of it all the time, or you know, you're just gonna make it slowly? Because depending on which protein you're trying to make, sometimes you might need a lot of them in a very short period of time. Okay, in some cases you only need a little bit. Then you're not going to make as much. And even after you make the protein, you can have post-translational control. Right? We talked about how the vesicles containing the protein can go and bind to the Golgi body, where you sort them, you modify them, and you can repackage them. And those proteins can stay in the cytoplasm for some time before it is going to be sent out for use. Okay, so you can control the expression at multiple stages. All right, now we're going to work on mutation. We're going to work on mutation. Mutation is a permanent change in genetic sequence of an organism, okay, in the DNA. The changes has to be in the DNA. Okay. And if you change the genetic sequence, you might change the amino acid sequence as well. Okay, is not guaranteed. And I'll show you examples of where you can change the genetic code without affecting the amino acid sequence. As we know, protein functions is dependent on the order of amino acids. So if you change the amino acid sequence, it's likely you will affect the protein's function. But again, as you will see, is not guaranteed to happen. It's possible to change the protein sequence and have little effect on its function. Now, mutation can happen at any stage of the cell's life, but it's particularly likely to happen during DNA replication. All right. I told you last class, the mutation can happen spontaneously. There is nothing you can do to stop this. This is just like bad luck. Think about this. If I ask you to I give you an encyclopedia, and then I ask you to type out the encyclopedia. Okay? Let's say I ask you to type it out 5 times. Okay, every single page, every sentence, you have to type it out. Even with spell check on, it's almost guaranteed you're gonna make some mistakes, right? Okay, like if you're really good, even if you're really good, you might make like 1 or 2 mistakes because you're doing it for so long, and then there's so much to copy. It's just inevitable that you're gonna make mistakes, right? And that's the same thing with your cells. Every time your cell divides, they have to copy all the DNA, and mistakes are gonna happen. Sometimes you can pick up those mistakes and fix it. Sometimes the mistake just persist, and that's when you're gonna have a mutation. I also told you mutation can be induced because of environmental factors, maybe exposure to mutagens, to carcinogens, right? To UV lights. All these things can cause mutation to happen even when the cell is not copying its DNA. Okay, so these can lead to permanent changes in your DNA. So I'm gonna do the next part here on a note. You should definitely copy this, and you should have the table, the codon chart with you. Now, I cannot stress this. Okay, I don't know if you guys heard me last time. When you do the quiz, the next quiz, quiz number 3, and the final quiz, you have to have the codon chart. Okay, and you cannot have it on your computer because you're going to be using a lockdown browser. You won't be able to open anything else, right? So you don't have it on your phone. Better yet, print it out, have a physical copy.
Are these tables the same?
What's that?
The tables, are they the same?
All the tables are the same. Yeah. I posted another one on eCentennial. You can download it. If you are accustomed to using this one, this is at the end of your study guide, right? So make sure you print it out. All right. You don't want to be not having it, and then you see a question that needs you to use the table, and then you scramble to find it online and stuff like that. It's just going to be disaster. You have to print it out. Okay, you have to have a copy. Okay, let's take a look on mutation. Mutation. Okay, so there are different kinds of mutation. We are going to do the first one here called the base substitution. Base substitution. And this is where one nucleotide changes. Now, I want you to understand that the mutation happens at the DNA level. Okay, the changes has to be in the DNA for it to be permanent. If the changes happens in the RNA, it's not going to be permanent, right? The transcript is not going to stay there forever. As soon as the transcript breaks down, you can make a new one, and then no more problem. All right, so the mutation has to happen in the DNA level. However, to make it easier for you to understand, I am going to show you the changes in the RNA. Okay, so remember, the mutation happens in the DNA. But for easier understanding, we will just look at the changes in the mRNA. Okay. So if the DNA changes, of course, the mRNA is going to be different as well, right? Because when you do transcription, you're copying the wrong sequence. That's why it's going to be different. So this is the original. Original is called wild type. Let's say I have UUU. UUU. That's the original. Can you look up UUU again? You must be sick of looking up UUU. It's like my favorite example to use. What's UUU on the table? Yeah, top left corner, right? UUU is PHE. All right, so in the DNA, there was a mutation. And let's say it mutates to something. And then when we do the transcription, we end up getting UUC. Okay. Sorry, let me just reorganize this a little bit. This doesn't really look too good. This is the wild type. Okay, and then we have the mutant now. The mutant. That's actually what it's called. And it's UUC. Can we look up UUC, please? And this is the change, right? This is the mutation. What is UUC? Yeah, you guys just typing the same answer again, right? That's right. UUC. It's also PHE. So what's the result? What's going to happen if you have this mutation? What's going to happen if you have this mutation? Nothing is correct. Nothing's going to happen. Thank goodness. I want this mutation. It's going to do absolutely no harm to me. If I'm going to have a mutation, I want to have this kind of mutation. Okay? So no changes in amino acid. Okay? No effect. What do we call this? Type of mutation. Because nothing happened, we call this silent. You don't even know you have it. Silent or neutral mutation. Okay? It's the best kind to have. If you're going to mutate something, you better pray that you have the silent mutation. Okay? Here is an interesting question that I like to ask my class. You and me, okay? You and I, how much of our DNA are the same? Right? You and I. We are not related. I hope we're not. We probably different race as well, different ethnic background. How much of our genetic information are the same?
None.
None? Okay. 0. Anyone else? Let me repeat my question. If you look at all of your DNA, all your ACTGs, and you look at all of my ACTGs, how much of them would be the same? We have a two very polarizing answer here. Patrice says none, and Jay said 99, and then we got someone, Justine says 50, right in the middle. I love this. Okay, so it's very, very distinct answers, right? Anyone else? I just want to know what you guys think. All right, drum roll. Here's the answer. Between you and I, in fact, between you and anyone else on this planet, we are 99.9% identical on the DNA level. Isn't that crazy? Right? Only 0.1% difference between you and any other human beings, regardless of, you know, ethnic background, regardless of whether you are related to each other. Obviously, if you're related to each other, it might even be a little bit more closely together. But, you know, in general, it's 99.9%. How is that possible? Because we are all humans. Because we're all humans. Okay? Isn't that crazy? It's amazing, right? So that tiny, tiny difference and you're going to learn this in lecture 9, that tiny difference is a lot of times caused by silent mutation. Okay? Like you might have a UUU in your gene, and at that same location in my body, I have a UUC, and they both code for PHE. Okay, these single nucleotide differences, we call them SNPs. Okay, single nucleotide polymorphism. And we will learn more about this in lecture 9. Polymorphism is just, you know, like a difference in DNA. And knowing these SNPs is gonna help us do things like DNA fingerprinting. All right. Here's another question that blows people's mind. What about between humans and chimpanzee? How much of the DNA are the same between humans and chimpanzees? Throw me some numbers, guys.
It's gonna be like 98%, something like that.
98%, okay. Anyone, don't tell me it's over 90. Anyone else? Don't look it up on the internet. Someone this morning looked up on the internet. Give me a very precise answer.
75.
What's that?
75.
75. Okay. Yeah. Is 98.8%. Okay, 98.8%. If you look at all the DNA in humans and chimpanzees, and of course we are related because we are both primates, we are 98.8% identical. Mind-blowing, right? That small difference is responsible for the level of intelligence that are different between chimpanzee and human beings. Okay? Mind-blowing. Anyway, back to our regularly scheduled program. Let's try another one. Let's try let's say we have like, you know, AUA. Okay, let's look up AUA for me, will you? What's AUA? Yeah, AUA is ILE. Let's say I do a mutation here. Let's say I change the middle one to like C or something. ACA. Right? What is that? What is ACA? ACA is 3 only, THR. What's the result? Right. Well, now we have a different amino acid. Okay.
Sir?
Yeah.
When you're reading this chart, ACA, are you reading it left to right or right to left? Because I keep getting like
so this is the first, second, third. So you start with the first position. Okay, let's choose something that's not symmetrical. Let's say we look at ACG, okay?
Yeah.
So A, C, and then is this box. Do you agree?
Yes.
And then G. Okay.
Okay.
Got it? All right. So how bad is the result? Depends on where the mutation occurs. Okay. Type of mutation, we call this missense. Missense. It's a mistake. We made a mistake. Okay. Let me give you an example. Let's say this protein is an enzyme, right? You remember enzyme, don't you? Enzyme. Enzyme has these, let me try that again. Enzyme has these active sites, right? Active site. That's where you put the substrate in. if the missense mutation happens in an amino acid that is at the active site, that could be pretty bad, right? That could be pretty bad. That could cause the active site to change shape, and then the substrate will no longer fit. And you would cause the enzyme to not work anymore. But what if it happens like right here, right? This is like a part that's like not in the active side, it's kind of on the side, right? Then maybe it's not so bad, right? Maybe it's not gonna alter the function of the enzyme, right? So it depends on where it happens. Here is an analogy, okay? Let's say there is a super big nail, okay? Super big nail, and then it's gonna get stuck in your car. It's gonna get stuck in your car. Is that bad? Well, it depends where you stick it, right? If you stick it to the tire, not too good. The tire is gonna be have a hole in it, and then it's gonna get flat, and then your car, you probably, I mean, you can still drive it, but probably not like safely, right? Okay, eventually your tire is gonna be super flat, and you can't drive the car anymore. So that's bad. But the same nail, if I just like hammer it into your trunk, right? Is that gonna be bad? Yeah, I mean, like, it's gonna damage your car. It's gonna, not look too good, but it's not gonna affect the functionality of the car, right? You can still drive it, no problem. You don't even have to fix it, right? If you don't want to. So that's what's happening here. Even though we have a mistake, we have a mutation, we have a missense mutation, we have a nail, right? It depends where the mistake is happening. If it happens in a key part of the protein, then yes, we're gonna have terrible outcome. If it happens in like a part that's not really important, then maybe it's gonna be okay. Do you guys understand?
Yes.
Okay, thank you. Let's do one more, and then we're gonna go on break. Let's say we have CGA, and then I'm gonna mutate it to UGA. Right? It can happen to any of the nucleotide. All right, but the key thing is for base substitution, you are changing one nucleotide only. So CGA. Who was the student who was asking me how to use the table? Maybe you can show me this one so that I know you know now. I forgot who it was. CGA, what does CGA code for?
ARG.
ARG. CGA. Very good. ARG. And what about UGA? Anyone? What is UGA? Are you sure, Sarah? I don't think it's VAL. You want to try again? UGA. That's right, UGA. You got A, right? Remember I said you got A, you're awesome, you're good. That's the stop codon. Okay, what's the result? We have a premature stop. Okay, and you end up making a shortened. The proper word for shortened, like in biology, in this context, is truncated protein. Okay, this is a nonsense mutation. Why are you stopping? It's nonsense. You're not supposed to stop until the end, right? So if the mutation causes you to have a stop codon, that's a nonsense mutation. Now, again, right, like you're thinking, is this bad? Well, my answer is it could be bad. It depends. Again, it depends on where it happens. Let's say you have a protein that's supposed to be this long. Okay. If you have a nonsense mutation here, right, what's going to happen? Well, the rest of it is gone, isn't it? It's gone. Where's the protein? Not there. Is that bad? Yeah, that's pretty bad, right? Remember we talked about tumor suppressor gene last time? If you have one of these nonsense mutation and you end up not making the protein, then you have a loss of function mutation. Loss of function mutation. You can't turn on the red light if the red light is not there. We didn't even make the red light in this case. You can't stop the cell from dividing, right? So that's pretty bad. What if, how do I bring that thing back? Okay, right. Okay, what about it happens like right here? Is that bad? Probably not as bad, right? And you know, it might not even have an effect. Because if the nonsense mutation happens towards the end of the protein sequence, you are at the end anyways, right? Like you might lose a few amino acids and that might not have too much of an effect. Okay. My point is, you know, you can't judge whether you cannot judge whether one is worse than the other, right? If someone asks you, is missense mutation worse or nonsense mutation worse? What are you gonna say?
Depends
Depends on what?
where it happened.
That's right. Depends on where it happens, right? If a missense mutation happens at the active site, that's going to be worse than a nonsense mutation that's happening towards the end of the protein, right? In general, a nonsense mutation that happens at the beginning is probably going to be the worst, right? Because you're not making the protein at all, right? I hope that makes sense to you. So I'm gonna leave the screen on this page. You copy whatever you need to. We're gonna come back in 5 minutes, 1:45, and we still have quite a bit to do. Actually, you know what? While I still have your attention here, I just want to quickly talk about what's going on next week. Originally, we are scheduled to have the quiz on Tuesday. Okay, but judging from the progress from my morning class, I think it's best we postpone that, okay? So I'm gonna put the quiz on Thursday instead. I'm gonna post this on eCentennial later, but I just want to tell you first, okay? We're gonna move the quiz on Thursday so that on Tuesday we can practice more with this, you know, mutation stuff. And then on Tuesday, I will also teach you how to do the microscope lab. All right. There's some calculations, some math, some equations that I need to go through with you to make sure you have good understanding of it. And then no class next Thursday. Right? Next Thursday, you just use the class time to finish your lab to take the quiz. Okay. So come to class on Tuesday. We do more practice. We talk about the lab, and then Thursday you do the quiz. You do the lab. I will move the lab due date. I'll move the lab due date to Friday, so you don't have it on like both the quiz and the lab on the same day. All right? Okay, great. I'll see you in a bit, 5 minutes. Let's keep going, right? Because there are more mutations to come. But first, let me just go through the slides with you. These are just examples that like I was gonna do on the PowerPoints, but if I do on the PowerPoints, then I cannot post it afterwards, right? So that's why you know, I did the other note. This is an example that shows you the mutation happening in the DNA level. You can look through this slide yourself later on. I will be doing some examples with you, you know, with the changes at DNA in a bit, and that's probably more useful than me reading this slide to you. Okay, but you definitely should read this on your own after having a better understanding of it. Okay. This was sickle cell anemia. I first showed you sickle cell anemia. When did I show you sickle cell anemia? And the protein stuff, right? Like when you change the single amino acid sequence, this is a missense mutation, right? Only one change is enough to to cause the hemoglobin to misfold, and you know, it causes drastic changes in the way that your red cell shape, right? So next thing we're gonna do is another type of mutation. Let me just give you some background information first, and then we'll do some practice together. This is insertion or deletion mutation, as opposed to base substitution. In base substitution, we are changing a single nucleotide, right? It could be the first, second, or last codon. First, second, or third nucleotide in the codon. It doesn't matter which one you change. The key thing is you're changing one base at a time, right? One nucleotide at a time. For insertion or deletion, we are putting extra nucleotides into the DNA sequence, or we are removing nucleotides from the sequence. And when you do that, especially if you are doing it when you are inserting or removing nucleotides that are not multiples of 3, it will cause the reading frame to change. And we call that a frame shift mutation. You don't know what I'm talking about, right? I know, I don't even know what I'm talking about. So I'm gonna give you an example that will help clear things up, right? Just watch. Let's say we can only read sentences 3 letters at a time. Okay, that's the limitation. So let's read the first sentence. No problem. The big cat saw the fat dog. The sentence makes sense. I am reading 3 letters at a time. No problem. Let's take a look at here, right? In this first example, we removed the B, right? You see, we removed the B. So how is the sentence gonna sound like now? Remember, we can only read 3 letters at a time. That's the constraint, right? So the, no problem, same word. And then it stopped making sense. IGCATS, I don't know what I'm reading anymore. Right? By deleting a single letter, the entire sentence becomes gibberish, right? The entire sentence becomes something else, totally unreadable. We shift the reading frame. Okay? Does it always shift the reading frame? Do we always have a frame shift if we insert things or delete things? Not always. If you delete the whole word, if you delete multiples of 3, 3, 6, 9, so on and so forth, then the sentence structure remains unchanged. Right? Now I can still read it. The cat saw the fat dog. I lose a bit of information, but certainly not as bad as the previous one, right? Like this one is, I can't even understand it anymore. But this one, I still have the sentence structure maintained. Okay. So think about this. If we have a deletion in the DNA, if you delete 3 nucleotides at the same time, as opposed to 1 or 2, if you delete 3 nucleotides, what's the worst thing that would happen? You will lose 1 amino acid, right? But the remaining sequence will be intact. It's not going to have a problem, is it? Right? How about if you put an extra letter? You put extra K here. Again, the sentence structure is different now for the rest of the sentence. We have a frame shift. But it's okay if you insert 3 letters, right? 6 letters is okay. 9 letters is okay. I mean, you're still going to have a mutation. You might still affect the function of the protein, but it's not as bad as the other one, right? If you insert 3 nucleotides, you end up having an extra amino acid, right? If you insert 6 nucleotides, how many extra amino acids are you going to have? 6 nucleotides. If you insert 6 nucleotides, how many extra amino acids are you going to have? Not that many, not 26. Look, I'm gonna insert 6 nucleotides. A, U, U, C, C, U, something like that. How many amino acid is that?
2.
2. That's right. Because a triplet is an amino acid, right? So if you insert 6, you're gonna have 2 extra one. Do you guys understand? What if I delete 9? If I delete 9 nucleotides, how many amino acids are gonna be missing?
3.
3. That's right. 9 divided by 3 is 3, right? All right. We still have some work to do. So I told you to get the study guide, right? I hope you did, because we're gonna use it right now. And I want you to turn to this question 11. If you don't have it, that's fine. Just copy down the sequence. Right? But I hope you have it. It's much easier if you have it. The answer, by the way, is already posted online. You can definitely go take a look at it. But I want to work through this with you. So here's the original sequence, and I like to encourage students to chop it up. Okay, make sure you have some papers and pen when you do the test this time, do the quiz this time, because there's a lot of work that you have to do here. So this is the template strand that we are given. That's what the question says. And so when we write our mRNA, we have to do the base pairing rule, right? The complementary base pairing rule. So T is going to become an AUG. Okay, chop it up so we're not going to get confused. UGG, AAC. Please check my work. All right. Sometimes I make careless mistake, just like just like you guys. UGA. Now, for the interest of time, because this is old stuff, right? We learned this already last time. You look it up on the table. Okay? You can just believe me. You can just copy it, okay? And you can check it out later on your own. So if you look up AUG is MET. You should know that by now. That's the start codon, right? Start codon is always MET, MET, right? Methionine. And then the other ones is, I'm cheating here. I'm just looking at my chart. ASN, ARG, CYS, and then stop. Right? That's this is the original. This is the wild type. Wild type. The normal one. All right, I hope that's fine. That's like totally old stuff. Now we have a mutated CA sequence. We're gonna chop it up again. Gonna chop this thing up. And when you chop it up, we can see it's the same length as before, right? There were 5, 1, 2, 3, 4, 5, 6 triplets, 6 codons, and here we also have 6 codons, right? 1, 2, 3, 4, 5, 6. So I know nothing was inserted, nothing was deleted, and I have to find the mutation. Okay, so you just compare the letters one by one. TAC, TAC, no changes. ACC, ATC. Oh, I found a mistake. Right? There's the mistake. There is the error. So when I do my RNA sequence, I mean, I can just copy the original AUG, and then instead of UGG, because of the mutation, I would have UAG now. Okay, and then the rest would be the same. The rest would be the same. Now I want you to take a moment and do the translation for me. Write the protein sequence, please. Look at the chart. Look at the table. If you have it and you want to share with me, I should rephrase that. If you have the answer, I encourage you to share the answer with the class. What's the answer?
It's the same, but in the second one it's stop instead of the TRP.
That's right. So what's the first one? Can you tell me?
MET.
Yep. And then what did you
Stop.
say? Stop. Right. That's the stop codon, right?
Yep.
Thank you. Will there likely be an effect? Absolutely. Yes. This is for sure has gonna have an effect. Why? Because the protein is not made. Okay. So what kind of mutation is this? Well, it was a base substitution. You change a single nucleotide, and we ended up having a stop codon. So what do we call this kind of mutation? Is it silent? Is it missense? Is it nonsense? Like what is it?
Nonsense.
This is nonsense. Why are we stopping? You're supposed to keep going, right? This is nonsense. It doesn't make sense. That's what it means, right? Okay, this is a pretty bad mutation. You lost the protein. It's completely gone. All right. I hope this is making sense to you. I hope this is not nonsense to you, right? Okay, let's try the second sequence. What's the first thing you're gonna do? Gonna chop them up into triplets. Right? So you see, if I chop it up, I have a remainder or something, like I have an extra one. Right? So this is a hint that tells you we have inserted something. It's an insertion mutation. Right? Please find the insertion. What did we insert? What is the extra nucleotide here? Where's the extra nucleotide? Well, again, you compare, right? TAC. So we got TAC, and then ACC, but this is GAC. So there is the extra G. That's the insertion. Okay. So when you write your amino acid sequence, it's going to be, when you write your mRNA sequence, AUG, right? CUG, so on and so forth. Oops, not an A. And then I'm going to go ahead and do the translation. What do you think? You think there is going to be an effect here? Is this bad? depends on where, but you know where. It's right here. Is it bad to have the extra G there as opposed to, you know, have it at the somewhere else?
Oh,
No, it's not bad. Okay. Well, anyone else? Is this bad? How do you know if it's bad or not? It's pretty bad.
Yes.
Sorry, yeah, go ahead, Patricia.
I was gonna say, yes, it's bad because it doesn't give you the same protein.
Yeah, look, you were supposed to make this protein and then you end up having that protein. It's totally different, right? Because you shift the reading frame, right? You end up having something completely different. Yeah. It's like I order a cake, but then they make me I don't know. I just totally make that analogy on the spot. It's like I order a cake and then they give me a chicken. Okay. Like it's not what I ordered. Something totally different, right? You have a different protein, right? That's bad. It's pretty bad, right? Will there be a likely effect? Yes. Yes. It's a completely different protein. Okay. So we have the insertion, and that leads to a frame shift. Frame shift, the reading frame is different. It's like, you know, the red cat saw the fat dog thing, right? Like you take one letters out, everything is wrong afterwards. And what does the frame shift, what is the effect of the frame shift? Well, look, other than the first one, which is MET, everything is wrong afterwards. Everything is wrong. All these things are not the same as the original. We have extensive, like a lot, right? Extensive means a lot. Extensive missense mutation, right? And what's worse is the new frame, the new reading frame, the new frame does not have a stop codon, right? So like you just keep on making a random protein. It's super long and it's not stopping, and we end up having this chunk of protein that, you know, doesn't do anything. Okay. Doesn't do anything might not be the worst result. Okay. The worst result is you end up creating a protein that does something unpredictable. Okay. And it's gonna interfere with normal cellular activity. That could happen because like who knows what this sequence is gonna create, right? Anybody have anything you want to ask?
it's about like the test that you'll have like next week. Is it gonna be like this, or is it gonna be like multiple choice?
Oh, it's just all multiple choice. Yeah, yeah. It's all multiple
Okay.
choice. So like I could give you a sequence and ask you like what's the mutation, and then you will have to choose. Oh, this is a nonsense, missense, you know, whatever, right? It's not too bad. Don't worry. So I want you to try the rest yourself, and check the answers from online. Okay, I post the answer already. I want to quickly show you this one though. Okay, like I chop it up again. I chop it up. Right. Without looking too much into it. Just by chopping it up. Can anybody tell me what the mutation is? I'm not asking you where the mutation is. I'm asking you like what type of mutation is this? Like is it base substitution? Is it insertion? Is it deletion? How do you tell?
Deletion.
How do you know?
Short.
It's shorter, right? Like I'm supposed to have 6 of these things and then this one is like missing one, right? So I know something was deleted. That's why it's important. When you get a sequence, first thing you do is you chop it up. Right? Immediately you will know whether it's insertion, deletion, or base substitution. Okay, I have another one for you. Maybe I'll do it on the note so I can post it after. How do I go back here? There we go. So let's say this is the the coding strand. Okay, coding strand. Coding DNA. Let's say we have. Give me a second. I'm just trying to think about what to do here. ATG, CCT, AAG, CGC, CCC, AAA, TAG. All right. I am just going to quickly do the messenger RNA. Relationship between coding and messenger is that they're the same. You just copy them. All right. And of course, you can do the protein sequence, but never mind that. It's not important for what I'm about to tell you. Okay. So I am going to put an insertion here. I'm going to do an insertion mutation. And I'm going to do right here. Right here. Okay. I'm going to pop in an extra T. And again, why does this happen? It could be spontaneous. Just made a mistake when you copy your DNA. Could be a mutagen, right? That causes an extra T to go in. So if an extra T goes in, what's going to happen to your mRNA? You're going to have extra U, right? Aren't you? Extra U. So, ribosome. Ribosome, I'm going to read this thing. AUG. That's going to be MET. CCU. I don't know what that is. I got to look it up on the table. CCU is pro. And then what's the next codon the ribosome will read? What is the next codon? Correct, UAA. Right? Instead of reading the AAG, it's going to read the UAA. And what does UAA code for? Oh my goodness, wrong color. What does the UAA code for? Stop. That's right. You see? Insertion, in this case, can lead to frame shift. And frame shift can have extensive missense. Like the example we did in the study guide, right? And in this case, it could also lead to nonsense. If the new frame has a stop codon, right? So there are two ways to get a nonsense mutation. Well, three ways, really. You can have a nonsense mutation from base substitution. You can insert something and accidentally create a stop codon in the new frame. That's a nonsense mutation. You can delete something and accidentally have a stop codon in the new frame. Make sense? So many things could go wrong in the DNA. Come to think about it, we are quite fragile. All right, before I let you go, I want to do two more examples. Back to the PowerPoint now. Create a base substitution in the coding strand. This is coding. The question always have to tell you what strand you're looking at. There's no way to tell. Create a base substitution. That means changing one nucleotide, so that we are going to have a missense mutation. Okay. This is the easiest kind of mutation to do on an assessment. Okay? You can pick any of these that you like. Okay, let's pick this one. I can change any of the nucleotide. Let's say I change the middle one to C. Right. So let's take a look at the messenger RNA. The original, the wild type is AAG. And AAG, if you look it up, is lysine. And then the mutation, let's use blue here, change it to C. Again, you can change it to anything you like. You change it to G, change it to T, whatever. So the mutation is ACG, and ACG is THR. That's it. Right? Missense. You made a mistake. You have a different amino acid. And again, you could have done it like over here. You could choose this one, and I can change the CAG to, you know, AAG. Okay, so I'm just gonna do it here like AAG. Sorry, CAG. That's the original. CAG. It's glutamic acid. And then the new one, the mutated one, is AAG. AAG is lysine. Do you understand? As a teacher, this is very difficult to mark because all students are gonna, you know, potentially have different answers. But from your perspective, it's good because, you know, you have freedom. There's no fixed answer. Multiple correct ways to do this. Let's do one more. Create a base substitution in the coding strand. Okay, so this is again the coding that will result in nonsense mutation. So nonsense, that means we need to have UGA, UAA, and UAG. Okay, you have to create one of these things, right? So this is a little bit limited. You cannot do it anywhere. You have to find the right place for it. And it's hard to think in terms of DNA. So we're gonna change it into mRNA. And again, you just copy. Change the A to U, of course. So which nucleotide can I change so that I end up having one of these stop codons?
Can you only change one, sir?
Yeah, because the question asked for base substitution, right? Yeah, so like you cannot do this. I had students do this in like in-person tests before. They're like, oh, let's just erase the whole thing, and then change it to UGA. You are not allowed to do that because it says base substitution. You can only change one at a time. Is that what you're asking?
Yes.
Okay. Right, so where can we change? Where do we change? Where do we make the change?
At UAA?
The last one?
Yeah.
But that's already stopped, right? Like that's already a stop codon, and you are meant to stop at that point, right? Like when you make these proteins, you're supposed to stop there, right? So you won't be changing that. Blessing, you said CAG. Okay, I like that. And how are you gonna change that? Which nucleotide are you gonna change? The C, the A, or the G?
The G,
You're gonna change the G on this one? Okay, what are you gonna change it to?
A,
Okay, but if you do that, you are gonna end up having CAA, right?
Yes.
Is CAA a stop codon?
Yes.
It's not, right? These are the stop codons, right?
Oh,
You got A, you are awesome, you're good. That's how you remember it. So I need it to be one of these, but good try. RJ say, change the C to a U. That's right. Look, I can cross out the C and change it to a U, right? Because now if I do that, I have a stop codon. I have a UAG. UAG is a stop codon. Does that make sense?
Mm.
Yeah. Blessing, you understand that?
yes.
Okay. Can we find another possible location? There's another solution here. There's one other possible location where you can do a nonsense mutation. That's right, Abnet. AAG, right here, I can change the AAG to UAG, right? And then that's going to be a stop codon. But remember, when you do these changes, you must reflect them back into the original, right? Into the original DNA, like that. I think I forgot to do that in my, oh, I did. I did. Okay, so that's it. Lenny, last call before we go. All right, I know it's a lot of stuff. Okay, there are still a few more examples, 2 more examples here, which I will take up next class. And what you really need to do on your own is to work through the study guide. Okay, so there's this question that you have to do, but then there are a lot of other questions here, right? To create the mutation here, okay? And these tables, you should familiarize yourself on how to do these. Okay, and I'll take these up with you on Tuesday. Okay, along with the lab. You might want to print out the lab sheet so that you can take notes while I teach you stuff about it. Yeah, that's it for now. Okay, thank you for coming. Have a good rest of your week. Take care, guys.